Today. Dr. Michael Graham is coming to us from Madison, Wisconsin. It's a lovely place if you've never been. Can't recommend it enough. But it was really great to find another Badger to come talk to us today. And correct me if I'm wrong, but you are coming from a different point of view here because
I
you are an
am.
engineer.
That's correct.
And we are talking about blood flow, but from a much different perspective. And I think this is going to be really interesting to hear about because I don't think we think about what we do outside of a medical field. And I think it's really good to start looking at this and realizing how much more there is to it. And hopefully, this should give us a different perspective on something we probably take for granted.
All right.
With that, yeah, the floor is yours. And thank you all for coming.
All right. Thank you very much. So I really appreciate the opportunity to speak here, to speak in front of a medical audience. I also find it quite intimidating to do so. My background is I'm in the chemical and biological engineering department. Sorry, keep switching my screen here. I'm trying to get back to my gallery view. There it is. So I don't just see a blank, so I just don't see my screen. There we go. There we go. So yeah, so I'm in the chemical and biological engineering department at UW-Madison. I'm primarily a computational and theoretical fluid dynamicist, so a lot of mathematical modeling of various types of fluid flows, including blood flow. And so hopefully you will find my point of view on issues related to blood flow interesting. Just to get things started, I want to mention the people who did the work that I'll talk about today. A lot of it was done by Xiaopo Cheng, who just graduated from my group and actually is now a software engineer at NVIDIA. Del Zimmerman got his master's degree working on some of what I'll describe here. Some of the earlier work was done by Xiao Zhang, who won a poster award a few years ago for some of the work that he did here. We've been supported by a number of organizations in the past. We've had NIH support, NSF support. Our computational support comes from a large-scale NSF funded computing environment. And in the second half of my talk, I'll mention some collaborative work, a really important collaboration that we've had going on for many years now with Wilbur Lamb's group at Emory and Georgia Tech. So he's a physician scientist there. And we've also worked with a young, again, physician scientist there, Christina Caruso, and a little bit of what I'll talk about in terms of aggregation. Liz Ilfrig, again, a physician scientist there, have been involved with. And they've really kept us grounded and kept us cognizant of the kinds of issues that may be important in the biomedical environment. So just as some general background, things that people may be familiar with in terms of blood flow and the microcirculation from the physical point of view. This is a schematic of blood flow from an Annual Reviews in Fluid Mechanics article from Tim Seacombe. And I'll talk about a little bit about the mechanisms behind some of these phenomena later, but just as from the observational point of view, it's well known that in blood flow at small scales, there's a so-called cell-free layer near blood vessel walls, and the red cells tend to be somewhat set off from those blood vessel walls, whereas the white blood cells and platelets tend to disproportionately be concentrated in those regions. And the movie down here at the bottom, this is just showing leukocyte rolling in a rat cremaster muscle. You can't see the red cells; they're zipping by too quickly. You see white cells that are rolling along the blood vessel walls here. And then there's a phenomenon that goes along with the cell-free layer that's called plasma skimming. When you have bifurcations, then you're preferentially drawing plasma-enriched blood into those side vessels. So that's the plasma skimming phenomenon. There are important consequences of this non-uniform distribution of cells and these segregation phenomena. One classical consequence is the so-called Faraeus-Lindquist effect, which is just that if you estimate the viscosity of blood just from the Poiseuille's law formula for pressure-driven flow of a uniform fluid in a tube, using that formula will give you an apparent viscosity that depends on the diameter of the tube that you're taking measurements in, which should not happen in a uniform fluid. Poiseuille's law builds in the effect of the tube diameter in extracting the radius. And the reason for this apparent tube diameter dependence is because of the non-uniform distribution of the red blood cells in the tube, so that you have a lower viscosity region near the blood vessel walls that's essentially lubricating the higher viscosity region toward the core of the fluid. I mentioned on my previous slide that the white cells and the platelets are preferentially found near the blood vessel walls, and so that has benefits for immune surveillance and for hemostasis. Those won't be, I won't really be talking about white cells and platelets today. People have made microfluidic devices for separations of things like leukocytes, bacteria, circulating tumor cells, infected red cells, which are stiffer than healthy cells. So the picture here is basically exploiting this plasma skimming effect and also pulling off other types of other things that are driven to the blood vessel walls through this margination phenomenon. And then, of course, if you're putting artificial particles into the circulation, like drug carrier particles, then you can ask about what the effect of rigidity, shape, and size will be on the flow behavior and the segregation of these particles in the circulation. There's a nice review from Lola Eniola Adefeso's group from a couple of years ago. And then one thing that my group got particularly interested in a few years ago, again in collaboration with Wilbur Lamb's group, is how the cell distribution and segregation phenomena interact with the alterations in blood cell properties and interactions that might go along with various diseases. So, for example, sickle cell disease, but also other diseases, which I'll talk about later in my talk. This is a slide that may not be necessary in this audience, but we know that the different types of formed elements in the blood have different sizes and deformabilities. This is just a nice picture from a microfluidic high throughput screening device. This is a fairly old paper now, and many people do these kinds of things now, where they just looked at a measure of cell deformation and cell size, basically running cells one at a time through a little contraction expansion device that tends to deform the cells. And so we have the red cells, which are moderately sized and quite deformable because they don't have an intact nucleus. And so there's a big peak here, that this tail here of larger area, those are likely aggregates, cells that are stuck together. And then platelets and various types of white blood cells, these are much less deformable and much smaller than the red cells. And the size and the deformability turn out to play very important roles in how, excuse me, in how different types of cells segregate. Um, so we've been interested in these phenomena in the context of disease. I ran across a paper that referred to this paper from Van Leeuwenhoek. This is the Proceedings of the Royal Society in 1675, and I'll just let you read the quote. But I found it really interesting that, even with very primitive microscopes, here's a picture of one of Van Leeuwenhoek's microscopes. He was making observations about the behavior of red blood cells and how they seem to be stiffer than in disease than in health, maybe stickier. Quite interesting. I don't know what disease he might have had when he made those observations. I suppose there's a good chance it was just the flu. But I think this is kind of an interesting quote and ties in with some of what we'll talk about later in my talk. Okay, so let's just come back to more recent observations. And again, most of these are from a really fantastic Nature Communications paper from our collaborators in 2023. Just tying in, blood flow behavior to various diseases. So the aggregation behavior of red cells depends on whether you're healthy, or in this case, maybe if you have sepsis or COVID. This the top middle curve here. This is just an indication of the level of deformability of cells. Again, histogram of deformability for healthy cells, for cells from COVID patients, cells from people with sepsis. And so in these cases, the cell populations tend toward less deformable in these disease states. The picture on the right, let me just describe this picture a little bit. So this is from a microfluidic, essentially a blood vessel on a chip design. So these are endothelialized microfluidic channels that have then been dyed. And so the glycocalyx has been dyed. And so there's an image pre-perfusion on the left here and post-perfusion. And this was a specific case of healthy blood cells, but that had been put into plasma from a very severely ill COVID patient. So someone who had sepsis. No, sorry, this one is COVID, as opposed to non-COVID sepsis. So this study different types of blood disorders. And the fluorescence intensity is less in this case, indicating that there's less glycocalyx, that the glycocalyx has been damaged or in part removed due to the blood flow in this case. And then red blood cell shapes change in various blood disorders. And then I won't talk about this much here, but in sickle cell disease, for example, there are substantial changes in the morphology of the blood vessels in the brain, which may have important consequences as well. So I'll come back to how these observations tie in with what we've been studying as we go through the talk. So this is just kind of an overview. So we'll be interested in what are the factors that affect margination phenomenon, these cell migration phenomena that lead to margination, a little bit about mechanisms, and then what might the consequences be in this. Again, a lot of what I'll talk about here is work that's been motivated by work done by our collaborators. My group, we do simulations and mathematical modeling. And so that's primarily what I'll talk about in the first part of the talk. In the second part of the talk, I'll talk about I'll talk about some of the work that our collaborators have done, and how our simulations might tie into some of those observations. So the first work that we did on this was quite a while ago, and what we did at that point in time, this was from 2011. We made a very simple simulation study. So we have computational methodologies that allow us to simulate a fluid flow with particles in the flow, and the simplest case that you can imagine here that's somewhat relevant to the blood flow situation is we just have tiny spherical water balloons. So they have fluid inside them, they have elastic membranes, and we can make all of the particles spherical so that we control for shape, and we can just look at the effects of, for example, stiffness and size of the different types of particles. There's an important characterization of the properties of a flowing suspension with deformable particles. That's called the capillary number. Without going into the mathematics of it, the capillary number is an estimate of how much the viscous stresses are tending to deform your cells compared to how much the elastic properties of the membrane are tending to keep it at its rest shape. And so a low capillary number is a situation where the cells are essentially rigid relative to how fast you're shearing the fluid, or how viscous the plasma is. So it's a relative effect. So you're building in the issues of size and stiffness and viscosity and shear rate into this one quantity that gives you a ratio between the important effects that are going on. In all fluid mechanics problems, the other, oops, getting ahead of myself here, the other important quantity is the so-called Reynolds number. The Reynolds number measures inertial effects relative to viscous effects in the microcirculation, so blood vessels that are 100 microns or smaller. The Reynolds numbers tend to be much less than 1. The flow is laminar. You don't have development of strong vortices. You don't have development of turbulent flow. So that's the parameter regime that we're going to be in. The other important parameter from the blood flow point of view is hematocrit. And so in the fluid mechanics literature, that's the volume fraction phi. So phi in our nomenclature is hematocrit. All right. And then we can talk about different kinds of mixtures, how much of different components, stiff, flexible, large, small, we put in the mixture. So without dwelling on this, I just want to show a few essentially numerical experiments that we did. And these are very simple flows. So you can imagine a situation, we have a flow between two parallel plates. Top plate is moving to the right, the bottom plate is moving to the left. In this picture here, all the particles, all the cells are the same. They're the same size and the same rest shape. They're all spheres. But we've now sheared these, and the blue particles, which are stiffer than the red particles, they're now localized near the walls of this channel. You can see that they're all set off from the walls of the channel. None of these cells are touching the walls, but the blue ones are concentrated close to the walls relative to the red. So this is a suspension where we have mostly flexible particles and a relatively small concentration of stiff particles. So this would be similar to blood, where the red cells are the dominant component and white cells or platelets would be a trace component here. Though here we have many, many more. The volume fraction of the blue is much higher than you'd have for white cells or platelets. The blue, the case here on the upper right, this is a case where we have primarily stiff cells. And what you can see here is the relatively few more flexible cells are concentrated toward the center of the channel. So that you have margination of the stiff cells in a suspension of mostly flexible cells on the left. You have demargination of the flexible cells in a suspension that's mostly stiff cells. And then you can do the same things with small and large. So in this case, let me freeze this. You're seeing that the small particles are getting driven to the blood vessel, toward the blood vessel walls, when they're the trace component in a suspension that's mostly larger, flexible particles. So that's the basic observation. And one thing I'll emphasize here, this is entirely mechanical. There are no chemical interactions. There's no adhesion. There's no aggregation. There's no energetic preference of one component for the walls relative to the other component. If these touch the walls, they won't stick, for example. So this is a purely fluid mechanical phenomenon. We can go through and characterize that. One thing that's important to emphasize is that these segregation phenomena, these are characteristics of the mixture. If we just look at pure suspensions of the blue particles versus the red particles and look at how they're distributed across the channel, there's barely any difference. So this is just the number density of particles as a function of position in the channel. This is the wall. This is the center line. The stiff, there's always this so-called self-free layer here. There's a little bit of a peak at the edge of the self-free layer, uniform concentration in the bulk. This is simple shear flow, but very similar distributions if you only have one of the components. And then this is what the two bottom figures here are showing what happens if you change this is the number density for the stiff particles as you're changing the fraction of flexible particles. So as you make the majority component flexible, the stiff particles get driven right to the edge of the self-free layer here. Alternately, if you have the flexible particles and you change the fraction of stiff particles, as you make the stiff fraction dominant, the flexible particles get depleted from the bulk and concentrated in the center. All right, so that's this case on the left here, and then this case on the right here. So those are just some basic observations that we can make about when we have these mixtures of deformable particles in flow. I come from a fluid dynamics background. I'm interested in the fundamental processes that lead to these phenomena. So I won't get into all of the modeling details, but we can think about the key processes as just being twofold. So one is if we have a suspension of particles and they're being sheared, so they're driven so that they collide with one another in the shear flow. And you're coming back to this movie here. You're seeing here these two cells here. One is like sliding around the other, so there's a collision between those two particles. We can think about how much they get displaced in that collision process. And repeated collisions lead to a phenomenon that's called shear-induced diffusion. This has nothing to do with Brownian motion. It's just driven by the fact that your flow is causing these particles to collide with one another, and repeated collisions lead to this random motion. And the nature of these collisions depends on the size and the stiffness and the shape of these particles. The other phenomenon that's important is the phenomenon of deformability and the presence of a wall. And so there's a generic phenomenon. If we have a suspension of deformable cells, droplets, macromolecules in a flow, deformable particles, they want to stretch, the flow is trying to stretch those particles out, and they're trying to push back against the flow. And if there's a wall here, then the symmetry is broken. And as the cell is trying to pull back to its equilibrium rest shape, and this would be due to the elasticity of the cell membrane, for example, in the blood context, that cell will get driven away from the wall. And we have some understanding about that phenomenon, that migration velocity. And so these are the two key phenomena that are showing up in these suspensions. And so we can make mathematical models of various levels of detail that just capture these. And so here's, we can just think about, here's a collision between two particles, right, that have different stiffness. And in this case, so there's a background solvent here and a shear flow. I'm not showing the solvent, I'm just showing these suspended particles. And what happens in this case is that the stiffer particle gets displaced more than the flexible particle. So that plays into how those particles will tend to get distributed during flow. And then on the right here, this is just migration velocity as a function of how far you are from the wall. And the more flexible particles will migrate faster than the stiffer particles. In fact, for a rigid sphere in low Reynolds number flow near a wall, that particle won't migrate away from the wall at all. It'll just, whatever position you put it at t equals 0, it'll just stay there. And so we can make models that don't capture all of the details of the flow and the elasticity, but that capture these important effects. And I'm not going to describe the details of this this model, but Catherine, since you have a chemical engineering background, this is basically a model that has diffusion and flow in it. It's a convection diffusion or a drift diffusion model, and we can, in simple cases, we can extract some important features of the behavior from these simple models. And so let me just point out a couple things from that. One is that we can characterize the migration velocity. We can characterize these collisions with different parameters. And in the model, those all get absorbed into a quantity that we'll call M for margination parameter. And the most important thing about M is that there are distinct types of behavior depending on M. And in particular, if M is less than -1, the way we've defined it, then your trace component, your stiff particles, your small particles, your diseased cells, those will get driven to the blood vessel walls very, very strongly. And I'll show a little video of this in a second. And so I don't want to dwell on this, except to say that we can do some things like develop a master curve for the cell-free layer thickness in terms of the hematocrit and the channel size relative to the cell size. And that fits well actually with detailed simulations. It fits well with experiments. And then this margination parameter tells us about the extent to which the cells will, the minority component of the cells will get driven to the blood vessel walls or to the center line. So the little movie on the right is just, this is number density versus position. Here's the center. Here's the wall. The red, that's the concentration profile for the red cells, the primary component in this case. The blue is our initial concentration profile for our trace component. We just made it a step function profile here. And the main thing to observe is that this initial step function profile is going to evolve as we start the flow to a profile that's very concentrated, in this case, right at the edge of the cell-free layer. Here, it's a very simple model. There's no molecular diffusion, and our particles have 0 size. So they all end up in a spike near the wall. In reality, they'll end up in a little bit broader spike, but still localized near the wall. And I'll show you some of our detailed simulations of that point. in a minute. So the real punchline of these simple model results is that there are these important physical effects that, and in particular, stiffness and size that determine these segregation phenomena. And so with that, let me turn to how these phenomena might play out in the physiological situation. And so, so the first study that we did with any physiological relevance. So this is work that we did with Wilbur Lamb and his collaborators. And so so Wilbur's observation when he learned about our results was there are these observations in the literature of differances in effective white cell count for people who've been treated with corticosteroids, with dexamethasone and epinephrine. Those are the two that our collaborators specifically studied. And you could hypothesize that these differences in the white cell counts, they change on timescales that are much too short they change on relatively short timescales. And so one hypothesis is there's some biophysical phenomenon that's going on. And so what our collaborators did was just look at the effects of these drugs on the white blood cells. And they did this with healthy volunteers, basically treating them with dexamethasone or epinephrine, drawing blood, running them through these microfluidics and imaging where the white cells were. And so this is just a histogram of percent of cells marginated or demarginated. Just are they close to the wall? Are they not? And so this is the control case here and the dexamethasone case here. And what they observed was there's substantially more demarginated cells in the dexamethasone case than in the control case. And so along with that, they made the observation that these drugs alter the leukocyte stiffness. So they poked on these cells with AFM and measured an effective stiffness. That's what's shown on the upper left here. And then they also looked at cytoskeletal disruption after exposure to dexamethasone. And so here, these are cross sections. This is the control, and so you're seeing the bright green here indicating the cytoskeleton. In the treated, the cells from the treated patients, you see less. You see cytoskeleton disruption. So you see cells that are softer than they would have been otherwise. And that's completely consistent with our theoretical results that indicate that when you soften cells, they tend to have less tendency to marginate. And so this paper indicated that there's a potential mechanobiological role for some of these drugs in affecting leukocyte dynamics. So that was kind of the first attempt at a biomedical connection here. In our more recent work, we've started to think about blood disorders, and so I'll describe some observations again from our collaborators. This is again Wilbur Lamb's group, and so again they have these endothelialized microfluidics, and they've looked at various situations. So here I'll start with sickle cell disease. So they've flowed suspensions of healthy red cells and then various mixed mixtures of sickle and healthy cells through these endothelialized microfluidics, and then tested for pro-inflammatory signaling with these various dyes that detect various markers. And so this is just here on the right is normalized fluorescence intensity for two of these markers, and what they see is they increase the fraction of sickle red cells. They say increased fluorescent intensity. So the implication is that there's increased pro-inflammatory signaling. And I should mention that this this blood paper that came out last year, there's all sorts of additional work on transcriptomics. There's also a Nature Communications group paper that we weren't involved with from the same group with related observations. So very comprehensive observations about these endothelialized microfluidics. So sickle cells are generally smaller and stiffer than healthy cells. So by our theory and simulations, we would expect them to marginate. And so we can go ahead and make a computational model of our sickle cells, and we'll come back to that in a second. Let me just mention as well a study of again, this is from the Lamb group, where they looked at iron deficiency anemia and looked at a deformability index. This is blood from healthy controls and from individuals with iron deficiency anemia. So this is just frequency. This is just histogram of deformability. This is a joint histogram of deformability and area. And so you see two things here. So the deformability sees that you have a population that's shifted to smaller deformability. And this one, the changes in deformability aren't so obvious here, but the changes in size are pretty apparent here. So again, the iron deficient red cells are less deformable and smaller. And you can do, and our collaborators did similar studies with the endothelialized microfluidics and qualitatively similar observations. So increased pro-inflammatory signaling seen in the endothelium exposed to iron deficiency red cells. And then just in general, we can talk about other blood disorders. COVID leads to pathological cell shapes. So in particular, these spheroechinocytes, spherocytosis leads to stiffer small cells called spherocytes. And so in a paper that came out about a year and a half ago in Science Advances, we just made models, computational models of these four cases. So sickle cell, iron deficiency anemia, COVID, and spherocytosis. And the hypothesis that the work was built on was that in these cases where you have smaller and stiffer cells, that those cells will tend to marginate. They'll tend to get driven to the blood vessel walls. And because they're smaller and stiffer and present at the blood vessel walls, they're going to tend to generate large shear stress fluctuations that the endothelial cells will then be exposed to and respond to through pro-inflammatory signaling. And so with the computations, in experiments, you can't go in and measure the stress on an individual cell. You can measure potential consequences of that. But with the simulations, we can measure everything. And so this is what we did. So we did simulations now. These are no longer these spherical model systems. These are now somewhat more realistic. So we have biconcave discoids for our healthy cells. And then we have various types of disease cells here. We made some stiff curved sickle cells. Here in the iron deficiency case, we just made the cells smaller and a little bit stiffer. We have a model of spherokinetocytes. These are actually dodecahedra that with rounded tips that we made as our model of spherokinetocytes, and then the spherocytosis case. I'm not sure why that movie didn't play. Those are just smaller spherical cells. Okay. And so we can look at how they're distributed. And again, we can make little movies here. And so here, now this is flow in a tube. So the orientation is a little bit different. So the center, so the left is the center of the tube, the right is the walls. And so the red cells, there tends to be a peak, and this is observed experimentally. There's a peak in concentration at the center of the tubes, a little bit of a peak at the edge of the cell-free layer. And then here the sickle cells, you can see that they're concentrated in the cell-free layer. You see that in the movies as well. And the same qualitative features show up in all of these other cases. So in all of these other cases, the cells are different in size and deformability. They're also different in shape, and we've studied shape separately as a player, but it's really size and deformability that are the main qualitative players. And so, the shape is playing a role here, but essentially that's what you're seeing is these cells, the differences so in all these cases you see the concentration spiking near the walls, consistent with their smaller size, their smaller deformability. And then, in these simulations, we can just sit at a point on the wall and look at the shear stress that the endothelial cells would be exposed to on the wall. And so these are just traces of shear stress versus time for these various cases. And here we've subtracted off the average shear stress. And so the healthy case, you see little fluctuations, and this is the color coding here is the shear stress fluctuations. So you don't see any really big fluctuations here. The environment experienced by the endothelial cells in the healthy case in our model is reasonably consistent. But as you move to these other cases, in all these cases, you start to see spikes of high shear stress. The sickle cell case here quite distinctly, COVID case quite distinctly. Spherocytosis, less so. The small round cells, they just tend to go close to the wall and roll, and they don't generate big fluctuations. Oops. And then we can go in and just look at histograms of the probabilities of the shear stress for these various cases. Again, the red is the healthy, and then the various disease cases. And the real qualitative punchline here is that in these disease cases, the tail, the high shear stress tail of the distribution is much larger than in the healthy case. These fluctuations, these large fluctuations are essentially vanishingly rare if you're healthy, and become much more common in the diseased cases. And then the last thing, and I'll wrap up with this, is to look at the potential role of aggregation. And so part of this was motivated by observations from our Emory Georgia Tech collaborators that severely ill COVID patients have much higher levels of aggregation than healthy individuals. And so if you have much more let me just point out that the group, the dimensionless ratio that we care about in this case is the degree of aggregation, the force per unit area that's tending to pull the cells together versus the viscous stresses that are tending to pull the cells apart. We know that at rest, cells, even healthy cells, red cells, will tend to aggregate. But during flow, with any substantial shear stress, certainly the shear stresses that would be characteristic of the microcirculation, you don't expect there to be substantial aggregation if you're healthy. Whereas if in the diseased state, there might be substantial aggregation. So an aggregation parameter here of 1 would be that the forces pulling the cells together is comparable to the viscous stresses pulling the cells apart. So that's the test case that we chose. And so again, we can make these detailed studies. The one thing you see in the movies in the two cases is you see these big voids in the case of aggregation that you don't see in the absence of aggregation. And along with those voids, you see here, you can see actually this is a rouleau, a stack of cells that's forming. And what you tend to see is clumps of cells that now are closely approaching the walls, where in the case without aggregation, there's no clumping. Again, we can make some characterizations. Here's some cartoons of what we see, and we can make some characterizations of what's going on. And in particular, the average cell-free layer thickness is going up because the cells are attracted to one another. So you might expect that they want to clump and be further from the wall because they want to be close to one another. The flip side of that is the fluctuations become very large. So the mean cell-free layer thickness is thicker, but the fluctuations in the cell-free thickness are larger. And so that, again, when you turn to, and so this is just an image of that. So this is a clump of cells that's generating a very large shear stress fluctuation on the wall. And then again, in the beauty of simulations is we can go in and look at the shear stresses on the wall. And again, this is without aggregation. And with aggregation, again, you see these spikes here. This is an enormous spike in the wall shear stress that you would never see in the absence of aggregation. Again, we can quantify that with probabilities. And so again, here the tail of the distribution is much more substantial in the presence of aggregation. And then you can start asking the question is how does the aggregation interact with, for example, the presence of diseased cells? And those two phenomena actually tend to work together. And let me just show this probability distribution here. So this is, so blue dashed is no aggregation, no stiff cells. Blue is, this is stiff cells, but no aggregation. So you see a higher tail, high shear stress tail. Blue solid is sickle cells, or sorry, red dashed is, oh, I just noticed a typo, darn. Red dashed is healthy, healthy cells, but aggregation. And then if you have both the diseased cells and the aggregation, then you see that you have much higher probability of these high shear stress events than you did if you have healthy cells and negligible degree of aggregation. All right, so I guess in the interest of time, let me not say too much about the effects of what's going on if you now add the complications of geometric complexity. Now you start to have non-uniform distributions of the cells around the circumference here. If you have, for example, a tortuous serpentine channel. Let me just, in the interest of time, not say too much about that, except that now you tend to have aggregation, or not aggregation, you tend to have margination focused on this outer edge of the curved channel. So this is This is where the spike in the probability of in this case, sickle cells would be. We understand some of those things mechanistically. We can again look at the shear stress fluctuations. But let me just, interested in saving a couple minutes for questions, let me just wrap up with my conclusions here, and I would be happy to take any questions that anyone has. All right.
Dr. Graham, I think that Justin probably has remained in a detained state due to the traumas he was dealing with earlier.
Problem.
But he asked me to help close us out here. But very interesting talk. The modeling for the shear stress on those vessels in those different states is very impressive. We've got a bit of a mix here of trauma anesthesiologist, transfusion specialist, and I know earlier I saw Dr. Kirby on here, one of our trauma critical care surgeons. Any questions from the group for Dr. Graham?
Hey, good evening, everybody. This is Kathy Marcucci. So when Mike gave this talk to the NIH, as a transfusion person, keep in mind that there are 600 blood group antigens. When you're giving trauma blood, you're giving blood that's typed for 2. There are 600 antigens hanging off the red blood cell that you have no idea what they are. Some are proteins, some are sugars, right? And so, red blood cells that you're transfusing are very different than what's in the patient. And so sometimes you've got a cold patient, you're giving warm blood, you're giving cold blood. And so I think Mike's work is really applicable to sepsis or our low flow states. Sepsis, exsanguination, transfusion, microcirculation, that trauma that involves microcirculation. So I thought the group should know about this modeling, because I think if the sickle cell people can utilize Mike's work, we can as well, actually. And I showed this to John Waters. Actually, a little bit of this is in the book I'm doing. And John is like, I think this is why, we, a hematocrit, that's the crudest type of thing, right? And why our transfusion thresholds don't hold together, because what's going on in the microcirculation is very different than just checking the radial artery, and it's the crit is this. It's really, I think there's a whole subterranean world that can be informed by Mike's work, actually. I think it's absolutely fascinating, but he already knows that. So thanks, Mike.
Thank you.
I agree. Thanks for your comments, Kathy. Appreciate you joining us as well. Any other questions or comments? If not, Dr. Graham, thank you very much for contributing this month. I'm not sure, Aaron, do you know what Justin has on the schedule for our next trauma conference?
Sure. Let me pull it up here. Hang on just a second. So our next one is on Monday, June 16th, and that's going to be with Dr. Michael Kiersey. I'm not 100% sure of the topic yet, but yeah, coming up on June 16th.
All right, thank you, Aaron. Dr. Graham, if you don't mind, a lot of times we put up our multidisciplinary trauma conference presentations on the Trauma Anesthesiology Society YouTube channel. If you're okay with us sharing that, we would love to do so just to get it out to a broader audience. Perfect. Thank you so much. Well, thank you again for your contributions today. Much appreciated, and hope to see a bunch of folks at our next conference as well. Thank you. Have a good evening.
Thanks, Mike.